Hello students, in this video we are going to discuss in detail dark field microscopy. In that, first principle, ray diagram, working and applications. Principle of dark field microscopy. To observe the specimen in this microscopy, opaque disc is placed under condenser lens, due to which the light scattered by the specimen only reaches the objective lens, then to ocular lens for visualization. So in dark field microscopy, main principle and mechanism is presence of opaque disc. So this opaque disc, it is also called as a dark field stop. It is also called as a central aperture, and this opaque disc, it blocks the light rays, and there is no direct entry of light rays into the objective lens. Only the light scattered by the specimen enters the objective lens and then to ocular lens for visualization, and therefore we observe the dark background. And the specimen appears bright in color. So here is the image of RBCs which is seen in dark field microscopy. So RBCs appear brightly illuminated and the background is dark in color. Now the ray diagram and working of dark field microscopy. When we observe the ray diagram, we see here the light source, then dark field stop, condenser lens, specimen stage, objective lens and eyepiece lens. Working of dark field microscopy. In between light source and condenser lens, dark field stop or opaque disc is placed. Opaque disc prevents to fall the light directly on the objective lens. तो यहाँ पे dark field stop होती है, which is also called as opaque disc, and that dark field stop is present in between the condenser lens and light source. तो ये opaque disc का main function क्या है? It do not allow to enter the light rays directly onto the specimen and directly onto the objective lens, and therefore only the light scattered by the specimen gets entered into the objective lens, and this objective lens forms the primary image of the object, and final image is seen by the eyepiece lens of the microscope. So as light rays scattered by the specimen only enters the objective lens, the specimen appears brightly illuminated against a dark background. So here specimen appears brightly illuminated and background is dark. Here is the animated picture of syphilis bacterium. We observe the brightly illuminated bacterium against a dark background. Applications of dark field microscopy. Dark field microscopy is used to observe live cells and unstained specimens. So as staining procedure is not required for dark field microscopy, we can easily observe the live cells. Then to observe the morphology, motility and cell division. The external structure, external features of different kinds of cells, motility and cell division can be easily observed by using this dark field microscopy technique. Last application, it is used for observation of spirochetes such as Treponema pallidum for diagnosis of syphilis. So syphilis, it is one of the disease which is caused by the bacterium Treponema pallidum. So this bacterium, it is a spiral shaped bacterium, and this bacterium can be easily observed in dark field microscopy, and thereby this technique is used for diagnosis of syphilis. So thank you. If you like my video, please share and subscribe my channel.